Hi guys, this is our quick review of lecture 29, growth hormone for physio, or the physiology of the growth hormone. So we started off this lecture by going over the synthesis of the growth hormone. So before we delve into that, basically there are two primary hormones that are going to affect linear body growth. So you think of somebody growing taller throughout childhood and adolescence. So we have growth hormone, which is the primary one, and then growth hormone is going to stimulate the release of insulin-like growth factor, which is going to do the actual effect of causing somebody to grow longer, I guess is the correct way of saying it. And then growth hormone is going to have some metabolic effects as well. But before we go into that, let's talk quickly about the synthesis. So you guys remember we have the anterior pituitary and the posterior pituitary. We are concerned with the somatotrophs in the anterior pituitary, right? Anterior pituitary. So they're going to synthesize growth hormone inside of the anterior pituitary. They will store it in granules, and then they're going to secrete this growth hormone when they are stimulated to release it by GHRH, which is growth hormone releasing factor, which is going to be produced by a region of the hypothalamus. All right, so we're starting to see more of these nuclei or regions inside of the hypothalamus. Today we're going to be concerned with the arcuate region, which is going to give us GHRH. All right, so it turns out there's actually two forms of growth hormone in the body. They both come from the same pre-mRNA. There's the 22 kilodalton form, which is the dominant form, and then the 20 kilodalton form. And basically, the idea is that, as I said, they come from the same pre-mRNA. The introns get removed, the exons are left behind, and you get these two different mRNA molecules. They leave the nucleus, go to the rough ER, become a pre-pro hormone. Then they get modified inside of the Golgi and stored as are different versions of the growth hormone in secretory vesicles. And then eventually they will get released once the somatotroph has received GHRH from the hypothalamus. All right, but the lecture pointed out that there's actually growth hormone, although we only have two forms of growth hormone, it does exist as within a family of related genes. And so specifically, we were told that there are three that are very similar. So you can see homolog is like percentage that's the same in terms of structure, I believe. You can see that these all have the same number of amino acids. These four, which includes growth hormone, are coming from the same chromosome. And these three in particular, PVGH, human CS1, human CS2, are called placental chorionic somatomamotropins. And the idea is that they're helping with development of helping during pregnancy, so fetus, placenta development, etc. All right, so that brings us to what's going on with secretion, because we said that the growth hormones made in the anterior pituitary by these somatotrophs, but they're being stimulated to be released from the anterior pituitary by growth hormone releasing factor. So basically what's going on is we have the secretion of growth hormone under the control of two hormones from the hypothalamus. We have, as we've mentioned, growth hormone releasing hormone, GHRH, and that stimulates release from the pituitary. It's produced in the arcuate nucleus of the hypothalamus. Again, we are starting to learn about all these different nuclei in the hypothalamus. The arcuate nucleus has to do with growth hormone releasing hormone. All right, then we have our second hormone that's controlling the release of growth hormone from the anterior pituitary, and that's somatostatin, but I've heard people call it somatostopin, and so just as a good way to remember what it does, somatostatin is like a master inhibitor, and it is going to suppress the release of growth hormone. It's produced in a different region of the hypothalamus, so not the arcuate nucleus. Instead, it's produced in the paraventricular region. All right, but the idea is that the paraventricular region and the arcuate nucleus are going to release the somatostatin, SS, and growth hormone releasing hormone into the portal system, and that's going to take it to the somatotropes that are in the anterior pituitary. Once it's there, they're both going to bind to G-coupled protein receptors, slightly different ones, but they act on the same CAMP/PK signaling pathway. And the idea is that, so if we start with growth hormone releasing factor, it binds to its G-coupled protein receptor on the somatotroph, and it's going to, through its action, cause the conversion with adenylyl cyclase of ATP to CAMP. CAMP is going to cause pKa to phosphorylate these calcium channels and allow calcium to come into the cell, and then those calcium ions coming into the cell are going to stimulate the release of the vesicles that contain the growth hormone granules. Okay, so G-coupled protein receptor, adenylyl cyclase, CAMP, pKa, having calcium come into the cell. Then we have the opposite effect with our somatostatins, our SS. That's going to come in, bind to its G-coupled protein receptor, but that's going to inhibit adenylyl cyclase. And as a result of inhibiting sort of this early on step of the pathway that is going to release growth hormone, somatostatin is going to overall inhibit the release of growth hormone. All right, so not too bad. We just have growth hormone releasing hormone and somatostatin from the hypothalamus that are regulating growth hormone secretion. And of course, we know in the back of our head, well, we're also going to have some feedback mechanisms. So this is sort of like a before feedback overall. All right, so then other factors affecting growth hormone secretion, similar to our discussion of LH and FSH in the past, growth hormone is going to be released from the pituitary in a pulsatile fashion. And so you have spikes throughout the day. Here we see 8 a.m., 8 p.m., you can see some spikes. But in particular, you see that it spikes during sleep. So overall, sleep is going to be an important factor for the release of growth hormone. So one of the reasons why we say kids need their sleep so they can grow. And then one important thing to know is growth hormone has a relatively short half-life of about 20 to 25 minutes. This will change if it is bound to growth hormone binding protein, which we'll talk about in a little bit. And then another thing to know is the amplitude of the pulses of growth hormone decreases as you get older. All right, so other factors affecting growth hormone secretion, we have stimulating factors, energy deficiency, so hypoglycemia, not enough glucose in your blood, vigorous exercise. I think the thought is that strain on your bones is going to cause the release of growth hormone. And then fasting, certain amino acids, in particular eating a high-protein meal. And we were told the amino acid arginine in particular stimulates growth hormone secretion. And then testosterone and glucagon are also going to stimulate the secretion of growth hormone. And then finally, we've already mentioned this, but deep sleep, but not random eye movement sleep. Deep sleep that's not random eye movement, not REM. All right, factors inhibiting the secretion of growth hormone is growth hormone itself, right? It's the feedback mechanism, so negative feedback. Then insulin-like growth hormone, insulin-like growth factor, so IGF-1, which we haven't talked about yet, but that is also going to do negative feedback. And then too high of blood sugar, so hypoglycemia, and then a prolonged stress state, so continued high levels of cortisol will suppress growth hormone secretion, and then xenobiotics, which just means chemical substances that are foreign to the body. All right, so we're going to talk about the negative feedback overall, but these are good to know. All right, so then what exactly does growth hormone do? So growth hormone doesn't have an independent growth promoting activity, and we're going to see that we have direct metabolic effects of growth hormone, and then we have indirect actual growth effects of growth hormone. But the idea is that indirect, the actual growth that you think, the elongation of the human skeleton, is a result of this downstream hormone called insulin-like growth factor, so IGF-1. And growth hormone is going to stimulate the downstream production of IGF-1. How does it actually do that? Well, it activates another receptor, so GP130, so G-coupled protein receptor, and otherwise known as just growth hormone receptor, GHR. It's going to initiate this JAK-STAT signaling cascade, and JAK stands for Janus kinase, and STAT, we'll come back to that in just a second, but basically our growth hormone binds to the receptor, and we were told in class that cytokines and leptin can also bind to this receptor, so they're similar in shape. And then JAK gets phosphorylated, which you can see here. And then the receptor itself gets phosphorylated. And 1 second. After that occurs, you have STAT gets phosphorylated, and you can see that you have, after that occurs, then STAT goes into the nucleus, and it will start transcription of IGF and IGF binding protein. All right, but STAT stands for signal transducer and activator of transcription. All right, so basically what this is just saying is growth hormone binds the receptor. There's a JAK and STAT signaling cascade, and the end result is that you have IGF and IGF binding protein being produced. All right, so moving on, we have started to mention this, but we have the actual effects of growth hormone on the body. We have a direct metabolic effect, so it's going to cause the liver to increase the amount of gluconeogenesis that it's doing to produce more glucose. We have the adipose tissue are going to increase lipolysis, so give us more energy, and then muscle is going to decrease its glucose uptakes. All of these would be basically adding to the blood glucose concentrate amount, like concentration in the blood. So we would say these are all pro-diabetic effects that growth hormone has. And then the indirect effects are, well, we have the growth hormone receptors primarily in the liver, and so the liver is going to produce insulin like growth factor 1, and then that's going to cause the indirect effects of actual growth. And so IGF-1 will act on most tissues and organs, increase DNA, RNA, and protein synthesis, increase cell size, increase organ size and function, increase linear growth. All right, and then just more on the insulin-like growth factors. We actually have two different ones. There's IGF-1, which is doing all of this growth of the body, and then IGF-2 is not well understood, but the thought is that it's for fetal and newborn, aka neonatal growth, and insulin-like growth factors. So this is just a comparison of the polypeptide that is insulin and IGF-1 and IGF-2. Like, notice how similar they are overall. Right, section C is quite a bit different, but A and B between all of these, human insulin, IGF-1, IGF-2 are very similar, and then IGF-1 and IGF-2 are different based on 2 amino acids in this fourth section, D. All right, but IGF-1 and IGF-2 have insulin-like activities. Most IGFs are bound to IGF binding protein, which we've mentioned before, and the idea is that it prolongs the half-life, and then by having it bound to IGF binding proteins, you're going to basically blunt the physiological effects by releasing small amounts of IGF-1 at any given time. All right, and then just again based on this idea that insulin and insulin-like growth factors are very similar, the receptors are supposed to match that shape, so the receptors look similar as well. And so here you can see the insulin receptor. Here's the IGF-1 receptor. Right, notice that very little difference overall. And then the idea is that IGF-1 will bind to the insulin receptor and the IGF-2 receptor, but it has a higher affinity for the IGF-1 receptor. All right, and then we talked a little bit about IGF levels and the growth throughout age. Right, so people are familiar with spike in growth during puberty, but this is just expression of IGF and then rate of growth. So you can see at birth it's not as high, and then it increases and decreases with age. It has a spike at it during puberty. All right, and then this line here is growth. So we know that little kids grow really quickly, and then you have another spike of growth during puberty as well. All right, so then we have regulation of the growth hormone insulin-like growth factor axis. And so, we've already talked a little bit about this. We had, the factors that are inhibiting or suppressing. And so this is where we're just going to talk about the feedback a little bit. But remember, stimulating growth hormone, energy deficiency, certain amino acids, and then, of course, just, the very basics is going to stimulate the release of growth hormone from, like, a purely biological standpoint, right, the signaling cascade. But in terms of negative feedback, we have growth hormone and insulin-like growth hormone regulated by these negative feedbacks. Growth hormone in particular is going to inhibit the somatotrophs, right, these guys, from releasing more growth hormones. It's just like downstreams, like there's already a lot of growth hormone here, don't secrete anymore. All right, so that's the first step. So that's this guy right here. Inhibits its own secretion. And then insulin-like growth factor is going to inhibit, inhibit directly the release of growth hormone on these somatotrophs in the anterior pituitary, which we see here. That's this step. But then insulin-like growth factor is also going to act on the arcuate nucleus we talked about, and then the periventricular region to overall inhibit the release of growth hormone. And so the arcuate region, we said, is producing growth hormone, or growth hormone releasing hormone. So the idea is that insulin-like growth hormone factor 1 is going to inhibit the release of growth hormone releasing hormone from the arcuate nucleus. And then finally, insulin-like growth factor is going to stimulate the release of somatostatin, right, somatostopin. And so it is going, by increasing the amount of somatostatin, it's going to inhibit growth hormone production in the somatotroph, growth hormone production / release. All right, so that's just about it for this quick review, hopefully quick. We had two pathologies or categories of pathologies associated with growth hormone, basically just an excess. And so what we were pointed out to us was that, it really depends on the effects of having an excess of growth hormone, really depends on like when you have the excess. If it's during childhood and your bones haven't fused together, then you just become, very tall. And this is generally due to a benign pituitary adenoma, which just means like, mass, benign cancerous mass on the pituitary, anterior pituitary. But if it's after the bones fuse, then you get acromegaly, which is the deformation of the bones, coarsening with the facial features. And so the idea is like here, right, they're looking kind of, broader, I suppose, coarser, deeper voice. And then both of these types of growth hormone excess can be accompanied by it's excess growth hormone. Excess growth hormone is going to give you excess insulin-like growth hormone. And so if you have an excess of insulin-like growth hormone and that can bind to insulin receptors, growth factor, they can bind to insulin receptors, so you're going to get an insulin resistance. And as a result, it can be accompanied by insulin resistance. It can be accompanied as a result by type 2 diabetes. All right, then you could have deficiency of growth hormone or a defect in the pathway that growth hormone acts upon. So basically, if you just have low levels, that's a fourth to a third of dwarfism cases, but you have a proportional body in those types. And then if you have this defect in the action of growth hormone, example we were given is Laron syndrome, and the idea is that it's a form of growth hormone insensitivity syndrome, GHIS, and Laron in particular is a mutation on the receptor. But the idea is that growth hormone insensitivity syndrome can be the result of many different steps along the pathway. All right, so something wrong with the receptor, something wrong with STAT, something wrong with IGF overall, but different ways this can occur. All right, so we'll take a look at lecture 30 in just a minute. All right, so we are back with lecture 30 review. This is insulin and glucagon. I'm sure that you guys know a whole bunch about this. Insulin, of course, we know is, hey, cells, take up the glucose that's in the blood and go ahead and store it or use it. And then glucagon is, hey, we need some energy, output glucose into the blood, break down our storage, we need stuff now. All right, so that, let's just get started. So we have this first part is glucose homeostasis backgrounds. Basically, we're dealing with the pancreas overall being able to produce insulin and glucagon, and the idea is that we're going to try and maintain a blood glucose of around 100 milligrams per deciliter, and this is just done by maintaining a balance between uptake with insulin and then deposition of glucose into tissue, as well as getting rid of it with glucagon from the tissue. So glucagon is our signal of nutrient shortage. Open the stores, release our nutrients. Okay. And this is just a little bit of a diagram here. We have high blood glucose. It's going to the beta cells in the pancreas, and they're like, hey, we got a lot of glucose here. Could you please release some insulin? So the insulin's going to go out and tell the liver different signals, and go to the skeletal muscle and go to the adipose tissue. And specifically, we have the brain as insulin independent glucose uptake, but we are going to have this insulin go to the liver, adipose, and muscle. And so in the liver, it's going to say, hey, take up glucose, package and secrete our fats or triacylglycerols. The adipose tissue says, hey, stop breaking down fat. Go ahead and uptake this glucose in the blood. Start creating more triacylglycerols and uptake any free fatty acids that are in the blood, such that you can do this first one, increase triacylglycerol synthesis. Then in the muscles insulin's going to say uptake glucose, and then do some protein synthesis. Basically, use our high energy metabolites to make stuff, right? Anabolism. Okay, then we have glucagon. And glucagon is saying pretty much the exact opposite. We have low blood glucose, so liver only needs to come through for us and pump out glucose. And specifically, it works on the liver, kidneys, and adipose tissue. It does not work on muscle. And you think to yourself, well, if the point of dealing with low energy state is to remedy the fact that we're low energy and we want to give energy to the parts of our body that might need to, like pump our heart, like heart pumping, muscles moving, etc. You don't want to have glucagon acting on the muscles. You don't want our muscles to lose its fuel, right? So glucagon does not work on the muscle. All right. In terms of remembering the pancreas and its cells, this is a really dumb mnemonic, but it helps me. So you have bad PP cells, and so basically B for bad is insulin, secrete insulin. Why is this one first? Well, insulin's the first one we learned about. Then we have A, alpha cells, secrete glucagon. That's the second one we learned about. And then D for somatostatin, a sort of master inhibitor. And then PP, pancreatic polypeptide, polypeptides for digestion to break down different materials in the lumen of the gut. Okay, so how do we go ahead and regulate insulin release? We'll get to that in just a second, but it's just a reminder from our notes. We were told that the pancreas comes from the embryonic foregut and is therefore of endodermal origin. Is that a factoid you probably don't need to know? Yes, but there's a point made in the lecture to have that factoid. All right, so how do we go about regulating insulin release? Well, again, as we said, I'm going to hammer this point home. This is dependent really on food intake and blood glucose levels. So before you have food intake, so cephalic phase, insulin release is mediated by the parasympathetic nervous systems, PANS. And so basically, as you can see here, we have acetylcholine from PANS is going to act on our bad PP beta cells secreting insulin, and then you also have neuroepinephrine are going to decrease insulin. So that would be neurotransmitters effect on insulin secretion, so not too bad. Then we have, so now that you've eaten and you have a bunch of food in the gut, blood glucose levels are going to start to rise, but specifically, you have this food in your gut, and so there needs to be some sort of signaling pathway. And so we have the production of these hormones called incretin. And so incretin you may recognize GLP-1, so glucagon-like protein 1, and then GIP. And these are peptide hormones that are produced by L cells in the lining of the gut. So L, think lining, right? And it's secreted after food enters the gut. It's stimulated by, the purpose is, what it does is stimulate insulin secretion, okay? Then you also have, just pure, so this is like food in gut causes incretin to be secreted by L cells, which stimulate the beta cells in the pancreas. Then you also just have pure glucose in the blood, basically high levels of blood glucose, and you're going to stimulate the beta cells to release more insulin. All right, so more on that, glucose-stimulated insulin secretion. So here we have our friends, a nice beta cell in the pancreas. So the first step is to have glucose enter our GLUT2 transporter on the beta cell in the pancreas. It's going to, from there, stimulate glycolysis, right? How does that make sense? Well, glycolysis is the breakdown of glucose. So if you have a lot of glucose coming in, there's a whole lot of reactant, right? So it's stimulating glycolysis by its presence. You have an increase in ATP and ADH, and then your increased ATP and DH is going to inhibit the potassium channels in the cell membrane of our beta cells. If this gets inhibited, then you're going to have more positive in here, and the net change in the membrane potential is that you become more positive. This results in calcium channels being activated, causing calcium to come in. And so basically by closing the calcium channels, you have the membrane potential becomes more positive. That's going to cause depolarization and cause calcium to come in. If you remember our little action potential chart, and if calcium comes in, it's going to stimulate calcium release from the endoplasmic reticulum, and then even more calcium in the blood is going to stimulate the secretion of our stored insulin overall. All right, so may be helpful. I think the big picture is just insulin, glucose goes in through GLUT2, increases glycolysis, depolarizes the cell as a result, and as a result of that, you have a net increase in calcium, and then a net, the granules of insulin get excreted. Okay. Then we're also told that amino acids, in particular leucine, can also stimulate beta cells to release insulin. All right, so then we have our insulin is being told to be released by incretins, with food in the gut. You have a lot of glucose in the blood, and then we also have the parasympathetic reaction acetylcholine telling all these things are telling our beta cells to release insulin. Right, so food in the gut through incretins, aka GLP-1, high glucose levels in the blood, and then the parasympathetic nervous system. All these things are stimulating insulin secretion. All right, so then what does insulin do? We're going to go through adipose, liver, and muscle effects, but the major actions are it's going to stimulate sort of the positive pathways that are for building up things. Right, so you're going to say, let's do our anabolism pathways or build up pathways. Increase the uptake of glucose, fatty acids, amino acids, and then go ahead and store them as triglycerols, proteins, and glycogen. And then also at the same time say, hey, we're taking up glucose here, we're building stuff. It's we don't want to keep breaking stuff down, so we're going to antagonize or inhibit catabolism, those catabolic pathways, which are really our breakdown pathways. So we're going to decrease our catabolic pathways, glycogenesis, lipolysis, proteolysis, and then suppress hepatic gluconeogenesis. Right? We're also altering electrolyte balance, increasing potassium uptake and phosphate uptake, decrease sodium and calcium uptake. All right, but again, this is muscle, liver, adipose tissue are the primary targets. We're told a little bit about the insulin receptor itself. I think the basic thing to know is this is a surface receptor, and then it is made up of tyrosine kinase, and it's going to phosphorylate itself, and then cellular proteins that bind to the receptor, and we'll see that in more detail right now. So we were told that the insulin signaling pathway is a little bit different in each tissue. The idea is that when you have our insulin bind to its tyrosine kinase receptor, you're going to go ahead and have tyrosine kinase phosphorylate itself, but it's also going to phosphorylate this guy right here, and this is IRS, which is insulin receptor substrate, meaning something that binds to the insulin receptor. Wow, okay, what a great name. So then insulin receptor substrate is going to activate the rest of the cascade, as you can see here. So you have basically you're going to go through and cause insertion of GLUT4 transporters into the membrane. You're going to go ahead and inactivate glycogen breakdown pathways, and then you're going to stimulate via inactivating glycogen synthase kinase. So this guy, by inactivating glycogen synthase kinase, you're going to result in glycogen synthase not being phosphorylated and allow glycogen synthase to do its thing, meaning create glycogen from glucose. Okay, so IRS, insulin receptor substrate, P13K, ATK pathway. The basic idea is that you're going to mediate the effect on glucose uptake by activating your glucose transporters, getting them inserted into membranes, and then promote glycogen synthase by inhibiting the kinase that inactivates glycogen synthase. All right, so keep glycogen synthase from becoming inactivated. All right, overall metabolic effects you can see on this chart. So here we have primary effects of insulin. You can see increased insertion of glucose transporters into the muscle, increased glycogen synthase in the muscle, in the liver, increased the production of triacylglycerols, glycogenesis, increased glycogen synthesis in the liver, decreased gluconeogenesis, increased glucose transporters in the adipose tissue, and then increase the production of triacylglyceride in the adipose tissue. All right, but this is in line with hopefully what you're thinking. You just stimulate the anabolism pathways to build up pathways to store the extra energy that we've gotten from food. Right, so not too bad. More specifically on the liver, so these charts are a little bit annoying. I feel like this is a little bit redundant, but we're doing our best. So basically the idea is you want to suppress glucose output from liver by stimulating glycogen synthesis. And how do we go about and do that? Well, we have a whole bunch of different enzymes along the pathway to glycogen synthesis. But the first one is capturing our glucose. So we have glucokinase that we're going to go ahead and stimulate. Then we're going to stimulate glycogen synthase here to promote production of glycogen. We're going to inhibit glycogen phosphorylase to prevent the breakdown of glycogen. We're going to reduce the gluconeogenesis pathways. And so basically stimulate glycogen synthase, stimulate glycolysis, stimulate lipogenesis overall. And so how do you do that? Reduce 6G PAs, reduce PepCK, okay, then go for gluconeogenesis, reduce fructose-bisphosphate phosphorylase, so anything that is going to allow us to do gluconeogenesis. And then finally, we're also going to stimulate protein production and promote the synthesis and storage of fats, which we see here and here, okay, and then inhibit protein breakdown. All right, so this is a lot of repeating the same stuff. Adipose tissues, let's just look at the broad strokes. We have for recruitment, great, we use glucose to make triacylglycerides, and we're going to inhibit the breakdown of triacylglycerides, so inhibit hormone-sensitive triacylglyceride lipase, and then promote the production of lipoprotein lipase, and you can see that inserted here. And the idea is that if you're promoting the production of lipoprotein lipase, this is going to break down dietary triacylglycerides into free fatty acids, which can then be taken up into the adipose cell and converted into a storage form in the form of triacylglycerol. Okay, so then its effects on muscle, straightforward. We have insertion of glucose transporters. It's really the same in both muscle and adipose tissue, and you're going to promote glycogen synthesis overall. All right, then we're now on to glucagon. Remember bad PP. So bad B and bad is for beta cells, which produce insulin. A and bad is for alpha cells, which produce our second hormone we care about, which is glucagon. So alpha cells in the pancreas, they start as pro-glucagon, and then they get processed into glucagon in the pancreas, and then pro-glucagon is also going to be processed into incretin. And you remember incretin, or in the lumen of the gut, you had incretin produced by these L cells. So the L cells are taking pro-glucagon, and they are going to convert it into glucagon-like protein, and that will stimulate pancreas cells to release insulin. It's a little bit strange how that works out. All right, so then we have more on the production, regulation of glucagon secretion. This goes back to our blood serum levels of glucose. So if you have high blood glucose, amino acids, fatty acids, basically you're going to say we don't want to pump out stuff. If we have low glucose, so high glucose means don't pump out glucagon, we have enough glucose in the blood. If you have low glucose though, then you're going to want to increase glucagon. So glucose will inhibit, fatty acids will inhibit, amino acids will actually stimulate. So this one's positive, that's my bad. Then you have, this one's a little funny I think, sensory stimulation, so will increase glucagon secretion. So think there's a loud noise or you have pain, and so your body's going to respond to that by saying, hey, we need to make sure that we can deal with this, so how about you pump out some fuel from storage and let our muscle cells do what they need to do. All right, and then we also have alpha adrenergic signaling by the parasympathetic nervous system on alpha cells will cause increase in glucagon. All right, so parasympathetic stimulates, sympathetic will also stimulate, interestingly enough, and adrenergic are for sympathetic, and somatostatins are master inhibitors that will inhibit cortisol. Think about cortisol in this situation. Cortisol is a stress hormone, so you say, hey, we need to make sure we have enough blood glucose. All right, so then what does it do? I think we'll just go through this super quickly, because this is getting a little redundant, but we have primary effects of glucagon. Notice how nothing in the muscle, right? We don't want our muscle to give up its fuel. But here in the liver, we have an increase in gluconeogenesis, right? Use up our stores, output glucose to the blood. Same thing, stimulate glycogenolysis. We have too much, we need blood glucose. Stimulate glycogenolysis to output glucose in the blood. So overall, that will increase our glucose output. And then on the adipose tissue, we have all this energy stored in triacylglycerides. So why don't we stimulate lipolysis to output some fatty acids, which can then be used by the liver and convert it into a more useful metabolite, or you can beta oxidation and get our ATP more directly. All right, so then we had effects on the pancreas. Basically, we have a GPCR, G-coupled protein receptor for glucagon. And basically, just do exactly what we said. Stimulate gluconeogenesis, stimulate glycogenolysis. We want to stimulate PepCK, right? This guy. Stimulate the pathway that's for gluconeogenesis. So fructose 1,6 bisphosphate phosphorylates, and then dephosphorylating glucose phosphate, so G6 PAs. And then stimulate glycogenolysis by activating glycogen phosphorylase and inhibiting glycogen synthase. Okay, this is saying similar things. So inhibit lipogenesis and glycolysis, and then stimulate fatty acid oxidation to increase ATP for the cell, and then stimulate uptake of amino acids by the liver to be used for gluconeogenesis. Okay, moving on. So we had discussion of diabetes, type 1 and type 2. So type 1 is autoimmune destruction of beta cells. So we have antibodies against insulin or beta cell proteins. You have genetic predisposition. How do you treat this? With hormone replacement, unfortunately, or organ transplant, or decrease the body response to the beta cell. All right, type 2 is the one that is, if we have elevated fatty acids, say through obesity, mitochondrial dysfunction will follow, beta cell dysfunction, and basically you end up with glucose intolerance, and then you end up with pancreatic failure. And then more on the obesity side, if you have extra adipose, you can have adipocyte dysfunction, and this will cause an increase in insulin resistance. And then if you have an increase in insulin resistance, this will also affect the muscle. So insulin resistance to the muscle, less glucose uptake, so you have excess glucose in the blood. And so more glucose in the blood. On the liver side of things, you also have insulin resistance, so less insulin clearance, less glucose uptake, and then pancreas tries to compensate by pumping out an increase in insulin, but you already have insulin resistance, so it's kind of spiraling. Okay, and then adipose tissue dysfunction leads to lipotoxicity and insulin resistance. So basically, you keep taking in adipose, or taking fuel, make more and more adipose, and then if you have chronic positive energy balance, just like too much food all the time, then you'll have lipid spill over into the blood and cause metabolic disruption, which will then lead to insulin resistance. All right, so that's it. Exams tomorrow. Good luck, guys, and try our best.